thank you, and thank you for inviting me to come and give this talk. This is a beautiful view, actually. It's kind of distracting, and this is the first time I've actually been in an in-person talk. We've been in some in-person meetings, but very different looking out into an audience as opposed to the. But yeah, it's actually quite nice to actually see people in person as opposed to through Zoom. So I've got to see a few people that I have regularly. I wanted today to spend a little time about talking about the data curation efforts that we do within the All of Us program. You know, there are other aspects of my research around clinical operations and things like that, but today I'm going to really focus on the work that we've done in the All of Us program. Just to give people a little bit of context. So, okay. You know, the All of Us program was initially announced in 2015 under the Obama administration. It was initially started off called the Precision Medicine Initiative. And, you know, it was one of the kind of the last big projects that Dr. Francis Collins kind of initiated, and I would agree, it's one of the most ambitious projects I think NIH has undertook in decades, honestly. So, you know, the goal of the program, which I'll get into a little bit more, is to create a community of participants who are underrepresented in biomedical research and follow them through over time. So you kind of think of the Framingham study, but you know, present day version of it, where we're collecting a lot of different information. You know, the overall mission of the program is to accelerate health research and medical breakthroughs, enabling and individualizing prevention, treatment, and care for all of us. So, you know, that's the phrase of all of us is kind of embedded in this and representing of all of us in the U.S. And our general health. And so to that, you know, the three kind of goals of the program are to nurture an environment, collect as much data on these individuals who have participated, and, you know, create an environment, a platform to enable research on this data. You know, one of the big things about this program is that the participants are front and center. It's, you know, very participant driven. They're not considered subjects, but they're active members in the research consortium, which I think is very unique when we think about such a large program. You know, the goal is to collect 1 million, recruit 1 million individuals across the US. Right now, we are at 413,000 participants, and I'll get into details around like what makes a core participant and whatnot. 75% of them are represented in some underrepresented category, depending on how you slice the data. And, you know, 45% are from racial and ethnic minority groups. Which, yeah, if those of you who might have been, well, I'll get into that later. Just as a current state of the data set that's publicly available. When I say publicly, it's accessible by the researcher workbench when you go through a series of steps. But this gives you a general sense of a breakdown of some of those numbers I showed before, right? 24% are individuals over 65. We have about 25% where their annual income is less than 25K. You know, these are groups that might not normally be in research projects, and I think there's a whole piece that I'm not getting into around, you know, how do you engage these individuals in a research program? I know Lucella, when she was at UCSD, was part of the provider organization groups where they were recruiting sites, and I think you guys are still doing that or trying to onboard now at Yale. But we started off very small. We actually initially only had like 2 or 3 data types, but now we're at, we collect surveys, physical measurements, which are part of the kind of the core protocol, genotyping arrays, electronic health records, which I'll go into more detail, and that's where I spend most of my life in. Whole genome sequences, Fitbit data, they'll grow to other wearable devices, and structural variants and long read sequences, which are kind of new for this year. We're planning another release of this data, but this is a snapshot from earlier this year. I shouldn't say this year, we're in 2024. In about a year ago, actually, in April. But we're planning to do another release in, I think 4 or 5 months, where we'll have an update of all these data types. But when we first started the program, it really was just surveys and electronic health record data and physical measurements. So really, these 3 data types, and over the years, we have grown to a larger set. And one of the main missions of the program was to get biosamples and get the genomic data, which is very unique. Just keep an eye on time. This is just a brief review of how a participant gets engaged in the program. They learn about the All of Us research program through various mechanisms. There's recruitment sites, there's direct volunteer groups where they can find out through websites or ads that they see. They create an account, and in the process of creating an account and saying they're interested in the program, they give informed consent, and that consent, there's a primary consent. There is the EHR consent, which is saying that they are willing and acknowledge that they will be sharing their EHR data beyond what's just at their local recruitment site, which is actually kind of key for what the program is trying to do, which is trying to get the full picture of one's health. And so by consenting for that, they're also consenting that the program can go look for health information on that individual at other healthcare providers. And then finally, the genomics return of results consent, which is a whole process in of itself and how that consent process happens, but I won't get into it. Third step, I should say, is contributing data. They have to fill out 3 base surveys, which I'll get into a little later, and collect physical measurements. So they've consented, and they do these set of surveys and provide some biosamples and physical measurements, and that's considered a core participant. And then subsequent to that, they can actually view their data. So they have return of results on the genomic side, which I unfortunately won't have time to go into, and then also participate in other research studies. So the All of Us program is involved in other ancillary studies, one of them being the Nutrition for Precision Health, where participants that are already there can be recruited into those studies. But again, very focused around the participant. These are the main building blocks, and actually there are a couple of new blocks that will be added this year. But the main ones as of right now that are operational are the Participant Center, Participant Technology Center, which are definitely participant focused, the communication and engagement partners who focused on comms essentially, and the biobank, which is run out of the Mayo Clinic, and health provider organizations. These are the organizations that are actually recruiting the participants. And finally, the genomics partners, where all the biosamples get sent to them and they do sequencing. And there's 3 genome centers across the country that are doing the sequencing. And all that data gets funneled into the Data and Research Center. And that is where all the data resides, and that's where I'm going to be talking about is how do we process all that data. The overall mission of the DRC, it's the main kind of PIs are Vanderbilt, Broad, and Verily, and under that, Columbia is subcontracted to kind of oversee EHR data with what I mainly do, but also help I co-lead the curation efforts as well. But the overall mission of the DRC is to propel the All of Us research program forward by establishing a comprehensive data platform that seamlessly integrates, organizes, and optimally utilizes diverse data sets. And so I think the key here is this program is very unique in the sense of there are other programs that are starting off of this, but if you think about when it started in 2015, trying to get all this data into one place is not a simple task. And so our three main aims is get the data in, organize it, and make it useful. And that's the objectives of the DRC as far as what drives us forward. So I'll get into the data ecosystem that we call it, because initially, you know, it was just a few data types, and it's grown quite a bit, and the complexity of the program has grown quite a bit. So this is a high level view of how the data flows. We get EHR submissions, we get physical measurements are captured through this system called HealthPro. You have the participant portal where the surveys are coming in through. And then I mentioned this ancillary study. So the Nutrition Precision Health ancillary study has gone live. They're recruiting people, and we're starting to get data into the DRC from them. And all of that lands in what we call a data lake, or we call the raw data repository. This is where these various data streams drop the data on some cadence, depending on the data stream. And I broke this up into two groups because I think when we think about research, we think about, okay, we're curating this data set, but there's a whole other operational arm when it comes to data. And that's, you know, the metrics and the QC of the data, or just operational reporting that you might need to do for the program. So there's the curation efforts that supports research, and there's this curation efforts that supports operations of the program, both internally at the DRC as well as the program level. This is just a view of the kind of components of the general curation system. You know, we have, again, data coming from the EHRs, we have the surveys, but each one of these kind of boxes are a team, if you will, that are looking at specific data types. All of that kind of feeding into this curation pipeline, which is kind of our new focus this coming year within the DRC, mainly because we're trying to create this data set that has these multiple tiers. And again, we're getting all these various data streams, harmonizing it, and we're putting it into these 4 tiers. We have which we all call the curated data repository. So even though it's not one physical database, if you will, there are actually tiers of these databases. There's the public tier, which all of us can log on right now to data browser and researchallofus.org, and we can actually see general characteristics of the data that's been submitted to us. There is some cleanup and obviously generalizations that happen there. We have the register tier, which requires individuals to register, create an account as a researcher, and there they'll have access to de-identified data. It'll include surveys, EHRs, physical measurements, as well as some wearable data, which is the Fitbit data that we get. Another tier above that, which started in 2022, is the control tier. And this is relaxing some of this privacy and security rules that exist. Because as you start combining more and more data, the risk of re-identification increases. And that's actually a big thing that the program's thinking about in this coming year, because there's a big, let's say, component or building block that called CLAD. It's a data linkage group. And once you start linking a bunch of other data, the privacy and security re-identification issues start really popping up to the point that you might not be able to have a tier without some type of policy around having a DUA or some like review of the research itself. And that's still being fleshed out, and I'm sure like Lucilla is probably part of some of those policy designs as an advisory member. But the control tier is when you get to see some of the genomic data. And we relax some of the demographics as well. Because in the controlled registered tier, even though the objective is to recruit those underrepresented biomedical research, we do a lot, for privacy reasons, generalize a lot on the race and ethnicity. And that some of that gets relaxed in the control tier. And in the future, it's individual biospecimen participant data that will be collected, and that's TBD as far as when that'll happen. But for future ancillary study of NPH, where the researcher will have access to it, will probably be sometime next year. Actually, I think internally within the course consortium, it will be released for the consortium members, mainly the NPH group, but to the larger research community, I think there's still discussion there. Yeah.
So that will also have RNA-seq data, single cell RNA-seq data.
Yeah. And I'll get into that a little bit.
Oh.
And I'll be reading off a slide actually, because the genomics data is not actually my forte, but I think, the Broad Institute has been mainly handling all the genomics, and they've done a pretty amazing job as far as optimizing the data pipeline, I should say. So, kind of reiterating what that registered tier is, researchers get access to it. Essentially, it's an OMOP database. I was told Patrick Ryan gave a talk a few months ago on OHDSI, so I kind of just removed anything around OHDSI and OMOP, assuming you guys were familiar with that. But all this data gets into OMOP, and I'll go into that in later details. Again, the other thing I didn't mention in the control tier is we added like COVID EHR data, because it being a newer disease, we had to remove that in the registered tier because of risk of re-identification. But the general idea there is expanding on these data types so that researchers can leverage the data and start combining it. What we are seeing though is, most researchers handle one or two data modalities, but as they get more and more comfortable, they're starting to combine more. But before I get into the weeds of the curation process or the data types, I wanted to kind of step back and give a view of what's happened over time. So officially, we kind of started, even though if you think about it, in 2015, President Obama announced the Precision Medicine Initiative. The DRC started only collecting data in 2018, right? At that time, this idea of FedRAMP or FSMA, moderate environments in the research world was fairly new. And when I mean new, the concepts weren't new, but actual implementation of those environments were relatively new. There are probably one, maybe two other networks that were in that realm. But I think this was one of the kind of the first efforts where we were trying to do a lot at once. And part of that was a learning curve. We're on the Google platform, so that our cloud vendor is Google. All our tools run on Google. And that's predominantly because a lot of the genomics tooling has been built on top of GCE, or GCP, I should say, from the Broad Institute. But when we first went live, there were a lot of things that weren't FSMA compliant, or they hadn't gone through that security provisioning step. And so we were kind of limited in the tool stack that we could use. And I remember telling Josh at one point, we've written all this code. My hope is I'll throw this code away because there are things I want to use, but I can't because of the current security constraints. And so over time, we've evolved. We've gotten better at working through security reviews. There is this thing called the concept ConOps that we call it, which is like a document for that the security group, both internally and the NIH reviews before we can actually say we're going to do this thing. So Hua and I worked on a ConOp to get CLAMP running so that we can run the NLP pipelines. And usually it would take 6 to 8 months to get that review process done. A lot of it is education, but a lot of it was also bandwidth. And knowing the right things to say to the security groups so they could approve things, both at locally within the DRC, but also within the NIH. So now, we're a little bit more sophisticated in what we can do, the technology stack that we have. It took a long time for us to even have dashboards, which I'll get into at the very end, right? These are things that you're like, okay, well, I can hit the ground running. And one of the hats I wear is an operational role at Columbia, overseeing clinical systems and also helping with the hospital analytics. And so these things are kind of commonplace in a lot of organizations, but to get something set up in that type of environment takes time. And that was one of those like learning processes that we had. Whereas now you see N3C, you see other networks popping up where they have that kind of built in, and that streamlined process of getting FedRAMP has shortened quite a bit. And there's a thing called ATO, authorization to operate, which is basically a several hundred-page document saying these are the things we're going to do. And I know Lucilla has probably done several of those. So to give you a sense of like at a high level, our pipeline, back in the day when we started, everything was a very linear fashion because that's, how we first started off, based off the tooling that we had. And so we had this very long process of like, okay, we're going to create a CDR, a curated data repository, and that would take over a month because there were all these various data types that we had. Now, what we're calling Curation 2.0, they labeled it as Placid, which is essentially, creating a data lake, but each data type has like a set of owners, and we're iterating on the data on a regular basis. A lot of it comes from what we did actually on the EHR side. So on the EHR side, which I'll get into, is we have a kind of a continuous view of the data, and we're constantly processing it every day. So if you say, hey, I need the latest snapshot of EHR data, we have that because we basically have a process that runs every 3 hours, looking at the most current submissions and also the data quality checks that come along with it. But adopting that paradigm within for the other data types has made it a lot smoother, and is the process of, and we're in the process of making the historical stuff to follow this more current architecture. And part of this is because the team is so large and different groups are doing different pieces of curating the data, if you will. So I'm going to go into what we do with each of these data types. Let's see. Okay. So the 3 core data types, and these are the first 3 data types that started, which was participant surveys, EHR data, and physical measurements. They all kind of flow into OMOP, where we take the survey data, the participant information from the survey data, like date of birth, their self-reported population category gets put in the person table. EHR data pretty much goes into these visits, procedures, conditions, and drug and measurements tables. The physical measurements that get collected also go into the measurement table, and most of the survey data ends up in the observation table. So that gives you a kind of a high level view. The nice thing about that is because we're using OMOP and leveraging the Odyssey tool stack a little bit, we're able to standardize on the data, and we've also added a custom vocabulary. So now if you go into Odyssey's vocabulary browser called Athena, you can search for these concepts, these AOU specific concepts within that tool, which can make it easier for researchers as they try to explore the different concepts. Though I will say there is a learning curve for any researcher who doesn't know like data models or terminologies. I think that's where most researchers struggle with, is understanding terminologies and how they can be used. Yeah.
And the clinical notes, where we go. Yeah.
I didn't have, it is there, but it's not available to researchers right now. So, no, because of the security risks there. I have a slide later, and that's what Paul and I have been working on with like running a key on the notes. But as of right now, the note data isn't there. The most we have, it's not useful data, because like we have for the de-identification of the privacy cleaning rules that we have, we scrub any text fields. So even the title of the note's not going to be there. But one of the things we'll be doing in the future is Claude's group has got a classifier essentially. By looking at the titles or the notes, we can actually classify the LOINC document ontology code, and that'll be informative for researchers. They'll just know, okay, well, it looks like a nursing note was written, or a renal consult note was written, but that's about it. But that's currently not in the data set. Just to give a view of like the different surveys, like I said, there's these first three, the basics, lifestyle, and overall health are the three main surveys that a participant fills out. That's what kind of gets them one step closer to becoming a core participant. So when we say, we have 413,000 people, those are core participants who have consented and provided the survey and provided a sample. There are a whole bunch of other surveys that have been added, and the goal really is to get these participants to fill out these surveys. And those of you who might do survey research know that like there is a tapering of like completion as you add more. The most recent thing has been the pediatric population. So that started this past year as a pilot, where they're having participants with children to be recruited. It's a very, very small group right now, but the goal is to then get that data in. There's a lot of nuances that the privacy group is still figuring out, because, for example, we remove day and month, so you only have the year of birth, but if there are less than one, what do you do? Things like that, but there are other kind of nuances there. So that's the surveys that eventually get into the observation table, as I mentioned before. And that's really like a question answer pair, essentially, is what we do. There's also another view that's kind of a wide table view for the researchers to have one table. Right now, we have Fitbit data. It's under the bring your own device. There was also a study called the WHERE study, where they identified a set of participants and handed them a Fitbit, I think Fitbit Charge 3 maybe, where they kind of informally consent to share their data from their Fitbit data. But the consent process is more implicit, because the way they do it is within the app, they say share it, so that initiation is considered the consent, because the participant is initiating the sharing of that data. So they get these, and can't see it very well, basically a JSON payload from Fitbit that lands into that RDR, that lake house, and then we essentially pivot that data. It's nothing sophisticated right now, where we take the kind of minute inter-day level data as well as the summary data, and there's essentially 6 tables there. I will say this is kind of a little bit of the Wild West as far as like coming up with a standard on how to represent this. I know Ida Sims has been advocating for a data model there, and I think it's the mHealth model. But the data is so large, and that was more of an interchange format, but to run analyses is a different thing. And I think that goes back to the kind of question around, do you use FHIR or do you use OMOP, which we can get into at a later time. But one, in my opinion, is a data interchange interoperability solution, and another one is used for analysis and research, honestly. So right now we get the heart rate data, the activity data, and sleep data, and we have them as 6 different tables sitting on as sidecar tables to the core OMOP database. Hopefully, we were hoping to get it this year. It will be in the next release, or where we'll get Apple Health Kit data as well. But right now it's just the Fitbit data, and all this data is flowing in through the participant technology center. So, you know, that first diagram of the different building blocks, anything participant facing is going to be going through that group, and which is actually changing in I think 18 months or so to another group. The genomic data, like I said, this is a little bit outside of my wheelhouse, but what I do know is they've spent a lot, the Broad Institute in particular has spent a lot of time working on optimizing their pipelines, partly because of cost, right? So they built this thing on top of Google, on top of BigQuery, which is, BigQuery is Google's database or big table. So if you guys have used AWS, their version is Redshift, Google's is BigQuery. It's not your standard relational table, a database, I should say, but it's super fast. And actually, that is what the curated data repository is implemented in, is on BigQuery. But the genomic variant store, what they call, is built on top of BigQuery. It's very, very efficient. They're able, to give you a sense of cost. Like prior to building it, it was costing them, 12 cents to run a joint call analysis, and now in this year it's 6 cents. The goal that the program's trying to do in the Broad is to run a lot of the pipelines that a researcher normally would run, so they're not having to reinvent the wheel. And so have that kind of intermediate data just sitting there for them to then leverage. But all using the Broad tools. The platform, which is the workbench, is underlying called Terra, which is, I think some of you already know, a tool jointly built between Verily and the Broad. Essentially, it's a Jupyter notebook running on GCP. But all the tooling there that they have is pretty tailored for that. But the interesting thing is, and I'll get into it a little bit later, is when we go through the process of creating the curated data repository, right? So you have people submitting samples, and so the biobank has it in the Mayo Clinic, and you have the genome centers that need to run sequences on it. How do we figure out which samples we should be running to have the most value for the data set? Because we're still catching up, but as they get faster, they'll be kind of in line. But right now, what we do is we say, okay, what are participants who have a decent amount of data? Let's prioritize those as far as getting sequenced, because we won't want to sequence someone who hasn't, maybe doesn't contribute. They consented for EHR data, but don't really have EHR data, for example. Well, you can see here, there's, around 9,000 individuals who have both genomic data and wearable data. When you think about adding measurements and surveys and EHR data, we're around 200,000, which is very, very impressive in my opinion. Has anyone used all those data types? No, but hopefully one of you will. But I think that's a whole area of research is how do we use this multimodal data? I don't think anyone really knows what's the right way. This is the question about centralized versus federated. We have a central database with all this data, and they're coming from participants from all across the country, and we're putting into one database, and there's all this kind of nuances on how care is delivered. So like the last thing is EHR data, which I'll go into more detail because that's what I kind of cover on the program. We cover about 57 sites. We collect their EHR data in OMOP format. And, you know, as you can see, you know, there's a couple of groups that are just starting to onboard. So Missouri and the Greater Plains Pecornet group just onboarded. So I need to add them to this graphic. But on the most part, this is where all the most of the participants are coming. You know, they are, as we start looking at direct volunteers, those are the individuals that aren't getting recruited at a health provider organization. We'll fill in the gaps. And the program itself is focused on looking at other like provider organizations. So most of these are what we call academic medical centers. There are a few federally qualified health centers that are recruiting individuals, and those are really the group that we classify underrepresented in biomedical research. But collecting all of that data into one, there are these kind of nuanced biases. Like we know we know the participants from Columbia, for example, will have a higher rate of renal disease because we recruit from the renal clinic. There's going to be the same thing with a different disease from a different site. And we're adding them into one database as if it's one data set. I think that question of how do you do that analysis afterwards is an unanswered one, or how do you do it well, I should say. But, you know, as I mentioned before, we get OMOP data from these sites. All right, I'm going to run through these because I'm running out of time now. And we get it centrally into the DRC. They're in OMOP already. We do some QC checks. And we also have like a very active EHR community. We have biweekly calls. The consortium, I'd like to call it, a very friendly and supportive. So they share queries. If, you know, for example, at Columbia, we've shared our entire OMOP ETL on Epic, but we put it on Epic's user web because they're very particular. Epic is particular about sharing queries, but we have a venue for them to like kind of share and disseminate their knowledge there. But the nice thing, which took a very long time, again, thinking about what's allowed FedRAMP wise and security, prior to that, we'd run these checks and then we would have to send out individual emails, which is very cumbersome when you think about 50 some odd sites. Now we have an automated process where we have a set of rules. We say, okay, if they pass this threshold, well, we'll just send off a Zendesk ticket, which is, you know, kind of like a ServiceNow ticket or a Jira ticket. It's just a ticketing system. But we have now a log of that, and we can now track it better. So if we start thinking about how do we view the improvement on data quality, we have a system, a better system to do that. And this is just a more zoomed in view of what we're doing in the data quality checks. But for instance of time, when we process all this data, the next day our dashboards update. So sites as well as the NIH can look and see, hey, where are sites regarding EHR data submissions? And are there ones that are not like passing threshold that they're needed based off of their requirements that they have with the NIH? But this is also a way for us to start saying, okay, well, there's some laggards here, but there are also sites that are pretty sophisticated. So let's think about like adding more interesting data quality checks, right? You know, because data quality is like boiling the ocean. And so we need to have, in my opinion, we need more fitness for use data quality measures, but that really connects to other like leadership or program level decisions to some degree, which we're starting to get. And then, you know, about notes. So we've started a notes pilot. Hua and his team have been looking at running CLAMP in GCP. We have 4-ish sites who have piloted. We're in the process of doing the annotations to evaluate how well the concept extractions will work. The first thing that got accepted as far as through our privacy review board was only extracting concepts, nothing else, right? So you have even drugs or measurements we ignore, we just look at the concepts and we just put them in the node MIP table. The hope is this will go in what we call off-cycle, the off-cycle release. So we have major releases and then we have off-cycle releases, which allow us to add new data kind of iteratively. And then as they mature, we'll bring them into the mainline curated data repository, but they'll be like this side table that sits there. And you can see this as a little glimpse. You know, we see a lot of telephone encounters. Maybe it's because of COVID and the sample that we got. But again, not to be surprised, the progress notes are pretty high as far as prevalence. And this is just looking at the title names. So nothing that sophisticated, but gives us an insight in what sites have. And we have, like, again, a dashboard that like helps us kind of review some of these things. And we use Tableau for that. All right, so last part is how does this data support operations? So when you think about curating data, it's not just for research. There is all these pieces of supporting analytics, reports that the NIH might need, or even internally for leadership, and as well as a way to support external members. So there are other program members that might want to be able to explore the data outside of the workbench. These are these are like maybe the NIH data science team wants to have a better view on operational data. So all of that gets put into what we call the program data repository, the PDR. So we have essentially three repositories from an workflow point of view. We have the raw data repository, which is getting the raw data. We have the curated data repository that's frontward facing for the researchers. And then we have the program data repository. And so that's what actually runs a lot of our, like I said, Tableau dashboards are there. So if you think about the EHR ops data, we get the data, we then move it over to the PDR, and that's where we run the dashboards. And the PDR specifically is, I wouldn't say de-identified, but there's no text fields in that. Like the raw data repository, if you have, like, for example, in the drug table in OMA, there's the SIG column, which is a free text field. The PDR will not have that. That gets cleared out. So any text fields get cleared out in the PDR because there's a larger group. Even though it's consortium members who've been approved by the NIH to have access, we do limit what gets seen there. And these are just examples of some of the dashboards. So this is like the program level dashboard that the NIH kind of looks at on a regular basis to see where how recruitment is going across the country. This is one for the genomics side of things, really looking at return of results in like ancestry data as well. Mainly focused within the program as far as them making decisions. So the program really leverages a lot of these dashboards, which, like I said, took a while for us to have that set up for the security, because of security reasons. So in the early days, it was the NIH, potentially sharing a screen with us and us manually running things because we didn't have that security environment to show these visualizations. Again, these are reports on actually user surveys of the workbench itself. So not only are we collecting data on like the data flows, but we're also collecting data from the users of the workbench and looking at, what are areas that can be improved on. There are like weekly office hours where we collect data, but there's also surveys that get sent out within the workbench to these users. This is just highlighting some of the dashboards that currently exist. And when I say each one of these boxes might actually have multiple sub dashboards in it, right? And all of this is being driven off the PDR. So, there's quite a lot when we think about how the program has kind of grown You know, and a lot of this is, you know, tracking AIAN from the, you know, the tribal engagement, which was which has been a back and forth for quite a long time. And I know we're finally at a place where some of their data can be included, but using dashboards to track some of that, it has helped the program. Great. I wanted to end off with some of the summaries or like lessons learned. I think I mentioned some of them along the way. You know, one, when we started in 2017, 18, I think learning the cloud environment was a learning curve. It wasn't a straight, okay, this is the mental model I have of computing. It should just work there. Even though underlying it is the same principles. Because at the end of the day, when you're spinning up like an app engine or whatnot, you're spinning up a VM, but those are all things you're not exposed to. So that kind of layer of abstraction does make things kind of difficult at times. But I think, you know, we've gone through it, and I think the general research community has also gotten more educated in that. You know, the other thing to consider is there are many stakeholders when it comes to a data type. So, you know, we have the participants, for example, but there are, you know, program level requirements for data, so that we have to make sure we're capturing all the metadata that we think will be useful for a report or even for research, and making sure we have the right people there. Because when we start modeling the data, if we don't have the right people in the room, we can make silly mistakes. I think the other thing is, you know, being flexible. You know, I brought up AIAN. There were times where we've had to do several hot fixes where we spent a month or two curating the data, doing the QA, QC. We thought we had the green light, and then it was like, no, we talked to the tribal communities. We need to remove all the AIAN individuals and understand the decision, but then it's going back and having to do that in our older architecture was much harder. And then making sure there's no leakage, right? So the program, there were times where decisions for policy reasons had to be made, but that actually also affected like release dates and stuff, and a lot of engineering time as well. This is a big one. The data quality checking should be aligned, right? And I think this is where the program is finally moving towards. So from day one, I was said, okay, we can provide data quality checks, but we can do that forever, and still there will be something wrong with the data, right? And so if we can prioritize at the program level, like big buckets of like, okay, we really want to push this type of like these type of studies. Well, that gives us a starting point of like, what are big like diseases we want to focus on. So what we did internally was we said, okay, well, what are the main diseases that the CDC has been focusing on? We'll use those as our kind of first cases and start creating phenotypes for that, to then say, okay, well, if you want to do COPD research, like maybe these are some quality checks that we want to have incorporated. So those are the things that we're working on right now. But prior to that, it was more of a reactive nature, because yeah, we might have fixed this, but we might have been ignoring your units, because I was like, all right, well, how many people really care about units in the measurement? But there will be someone who does, and then we're scrambling. Well, why haven't we thought about that? Well, we have, but we just had it lower on the priority list because we just didn't think it was a strong enough use case. So aligning that at the program level, I think is very key. In standards development, you know, coming from the Odyssey world where we're looking at where we want standardized analytics, which requires standardizing the data, aligning those things don't always fit for operational needs, right? We need to get something out the door. I need to create new concepts, but Odyssey has this particular cadence on releasing the vocabulary, or even getting buy-in with a larger research community that says, hey, why do we want to add this? Does it really make sense? It makes sense for us, for the program. And so aligning those things takes some time. And has been challenging. And I think we've made huge strides, but I think that is a factor that people don't think of that often. I just wanted to end off, just kind of give you a sense of like the community and provider partners in the network and all of us. It's vast, and this is actually a slide from 2021. So I will say many of these slides I got from other members within the DRC and the consortium. And again, these are consortium members. So these are people who are actively within the program, and they keep growing every year. So it's a lot of people, a lot of groups trying to coordinate their data, and it's fairly complex from when it first started. And I think understanding that and understanding how all of this translates into a final data set that you use as a researcher, I think as researchers, there's a class of people, they say, oh, here's the data set, and they think it's perfect and that's the way it is, and they don't actually question it. That shouldn't be the case. Like, if you know how the sausage is made, you'll know sometimes I don't want to eat it. But I think with that knowledge, you can actually make better analyses, I should say. So with that, I wanted to thank everyone and thank the DRC team, because I'm just presenting on behalf of a lot of work from a lot of people. So I'll open it up to questions. Thanks.